Hi guys, welcome to lecture 4.1 where we will be talking about the process of abiogenesis. So we're going to be talking about how monomers come about, how we get macromolecules, what a protocell is, as well as the appearance of self-replication. Now, before we get going though, I do want to remind you this content is not in the OpenStax book that you have been getting most of your skimming content from, okay? So it's kind of a byproduct of switching over to a new book. We still like talking about the process of abiogenesis. I think it's important, but the OpenStax book does not talk about it. So the figures that you're going to see in this next lecture and in lecture 4.2 are actually from Campbell Biology. This one right here, which I realize is backwards to you guys, but that's fine. So if you go to the 10th edition of Campbell, that's where you can find all the figures that I reference in this lecture. Now, that said, you don't have to do that. I give you all the figures in this lecture. You guys can watch the recording as much as you want. And yeah, so you don't really need to do the usual kind of reviewing and skimming of the textbook and all that fun stuff. You don't have to really worry about that for lectures 4.1 and 4.2. Because again, we're not using the OpenStax textbook. So just use the lectures. And if you really need to, you can Google search. I know it's obnoxious for a professor to say this, but you can just Google search the figures for Campbell's 10th edition. The book should look something like this, and you should be able to find the respective figures that I mentioned in the slides. That's it. Carry on with lecture 4.1. Let's take a gander at the lecture question from last lecture segment, lecture question 3.2, which the following are examples of polymers. Okay, so if you said D, you would be correct, and I'm not going to tell you why. You have to ask your neighbors or your friends or your grandmother, whoever you think knows the answer and can explain it, if you don't know the answer and can explain it. And it's really important that if you know the answer, you can explain the answer, right? Wonderful. Okay, let's dive into lecture 4.1. So to start you off with, interesting. So this is a photograph of the Danakil Depression in Ethiopia. So these are salt and sulfur formations and are very likely similar to the places where early life formed on Earth. So an early life, or the process in which life developed from non-living matter, is what we call abiogenesis. So before we get to the development of life, though, we need to have a couple steps that we have to cross. We need some 4 requirements that need to happen. The first of which is we need some monomers. We need the abiotic synthesis of monomers. Right? We just talked about monomers a minute ago, or a couple lectures ago, and how we can synthesize them. Well, now we need to be able to do that without anything around, right? So the first requirement for the abiotic synthesis of monomers is to have little or no free atmospheric oxygen. And if we remember why, well, if you think about why, let's remember our electronegativity in our periodic table of elements, right? Oxygen is extremely electronegative. So it tends to break bonds or oxidize. And if there's too much oxygen floating around, those monomers would just break down. So currently, there's about 21% of our atmosphere is oxygen. So if we didn't have oxygen at the start of all this, where did that oxygen come from? We'll get to that. All right. So first, before we can start abiotically synthesizing monomers, we need to have no oxygen. That's the first requirement. The second requirement is we need to have some source of energy. We need energy to start forming the bonds, start sticking those molecules together to form the monomers. There's a bunch of different sources that we could have for this energy, ranging from thunderstorms to volcanoes to meteoroids or even radiation, but no one really knows. But you definitely need to have some source of energy for this to happen. The third requirement is you need to have the kind of major building blocks, right? You need to have those chemical building blocks, which include the carbon, hydrogen, oxygen, and nitrogen, CHON, that we've talked about several times. A couple of different places that we could get those things. We could get water, dissolved organic minerals. Of course, the atmosphere contained a lot of that. Contained a lot of carbon dioxide and water and carbon monoxide, as well as hydrogen and nitrogen gas. So it's very plausible there was ammonia, hydrogen, sulfide, and methane that was actually delivered to Earth via comets. That's one of the prevalent theories, is that a lot of these initial building blocks were brought to us by comets. So, yeah, like that. The fact that we all came from outer space, that's not so ridiculous, just not in the way that people think. Anyway, okay. So that's where we can get our basic building blocks. The atmosphere, or maybe, depending on what they are, extraterrestrial sources. The last thing we need for the abiotic synthesis of monomers is we need a lot of time. So we have all the basic parts and the energy to stitch them together. We don't have too much oxygen to mess things up. So now we just have to wait for a long time. So Earth is about 4.6 billion, that's billion with a B, billion years old. And we think that life started about 3.5 billion years ago. So I'll do the math for you. That's about 1.1 billion years we had to wait for anything to happen at all. We just had to sit and wait. So, how did life arise then? How did this process of abiogenesis occur? Well, starting, of course, with the abiotic synthesis of monomers. Well, there's a couple prevailing theories. The first is the prebiotic soup hypothesis, put forth by Oprah and Haldane in the 1920s. And the idea is that the life formed near the Earth's surface from the spontaneous formation of monomers. So, seems pretty straightforward. And it just so happens that somebody actually tested this. Miller and Urey actually conducted an experiment that tested and supported the theory. Well, it ended up supporting the theory. They didn't know it would. So they created a sealed system where water vapor was pumped through a chamber filled with atmospheric gases like methane and ammonia and hydrogen gas, and had an electrode in that container, that chamber, that was zapping electricity through the chamber. That's, I think, the technical term, zapping. And then they condensed the water from that chamber back down into a liquid again. They sampled from that liquid, and lo and behold, they actually found some organic molecules, including amino acids. And amino acids are the monomers of what structures again? And what are the major functional groups that make an amino acid? And what are the bonds that stick those amino acids together? I'm not going to tell you. You have to go look it up because we just talked about it. So figure those questions out. What are amino acids monomers of? What are the major functional groups that make an amino acid? And what are the bonds that stick amino acids together to form the polymer that they make? All right, figure that out. Anyway, so that's the abiotic soup hypothesis. The other is the iron-sulfur world hypothesis. Basically, the theory goes that so there's these hydrothermal vents down at the bottom of the ocean that are spewing out super hot liquid and have a ton of materials and minerals like nickel sulfides and iron. And iron is actually a known catalyst for building molecules and facilitating the formation of those molecules. So it's very plausible that you have the energy source of these hydrothermal vents that's helping fuel these chemical reactions, and you have iron that's facilitating those chemical reactions, those initial building. Very, very plausible. Kind of a fun little side note that I'm going to try to do a little bit throughout the course here is throw in some references, some cool stuff around Rutgers. For example, if hydrothermal events are interesting to you, you can actually check out some really cool research going on about those hydrothermal events at the Rutgers and the Marine Science Department. Actually, the Center for Deep Sea Ecology and Biotechnology does some fascinating cross-disciplinary stuff with biologists and geologists and chemists and all those kinds of cool stuff. So you can check them out. That's the link down there, deepsea.marine.rutgers.edu. They do some really cool research if that's interesting to you. And I'll see if I can pull some other cool stuff that goes on at Rutgers about the topics we talk about. There'll probably be a slight bias, at least initially, because my background's in ecology. So I know a lot of the people and a lot of the some of the research that goes on over on around the ecology department and around the School of Environmental and Biological Sciences. But I'll start seeing if I can pull from other resources as well. We'll see how it goes. Anyway, so that's something you need to check out if you're interested in hydrothermal vents. Okay. So now we got these major building blocks of monomers, right? So how do we start snapping these things together? Well, it's actually a lot simpler than you expect. You could literally take some monomers and drip them onto hot sand or rocks, and the negative ions bind those monomers together. You can literally drip amino acids onto hot rocks and they'll form polypeptides. So what kind of reaction do you think that is? What do you think? Reaction that's taking the monomers and it forms them into polymers. What reaction? I'm not going to tell you. You have to go figure it out. Okay. So yeah, this is pretty wild. You can do this at home. Well, maybe not at home, but you can do this. It's been done. So now, we have this simple building blocks and some of the more complicated building blocks, but no cells yet. So what happens when you drop lipids into water? Remember we talked about that a little bit ago? Well, so these lipids can form vesicles, right? They'll automatically form vesicles. So a vesicle is basically just a kind of bubble where the outside, that kind of separates the outside from the inside. And so this will actually happen, and you'll form a kind of protocell, right? So these structures or protocells kind of behave a lot like a living cell. They do things like osmosis, which is exchanging water across this vesicle that's formed from the lipids. They can do some kind of regulation of homeostasis, where they try to maintain an environment that's different inside than it is outside, an environment that's more conducive to their life. They can even divide, right? So they can even replicate. But, and this is a huge but, they do not have any kind of heredity, right? They cannot pass on their traits to their offspring. And that's really critical differentiation between a protocell and what is a living organism. All right. So now what we have, along comes ribonucleic acids, or RNAs. So RNAs are really cool because they can actually self-replicate, and they can help catalyze protein synthesis via ribosomes, which are basically RNA molecules that can act like protein-based enzymes. So facilitating chemical reactions, right? Now, of course, everyone knows and loves DNA, but that came later. Really, the biggest advantage of DNA over RNA is that it's double-stranded, right? So you have two of these strands, so it makes it a much more stable platform for passing on your information. There's much less likely to have errors, right? So great. We were able to get our initial monomers. Let's kind of recap real fast. Right? We got our initial monomers after we had these 4 things that needed to happen first. Then we had those monomers were synthesized into macromolecules. Then we had a formation of protocells, which have this kind of bubble or vesicle shape. They're able to kind of do these things that are similar to living organisms: osmosis, homeostasis, and division. And then finally, we had self-replication or heredity. So we had RNA, which served a bunch of different functions, including catalyzing reactions and passing on the genetic information, and then eventually DNA. So now we have all the basic building blocks of life. There it is, done. Now we have, theoretically, living organisms. Cool. So that's it for lecture 4.1. We're going to move on now to lecture 4.2. But before that, let's take a look at a lecture question. Which of the following are required for the abiotic synthesis of monomers? Low levels of atmospheric oxygen, high levels of atmospheric oxygen, basic molecular building blocks, enzymes to facilitate dehydration synthesis reactions. Okay. Well, you can figure that out and figure it out before lecture 4.2, which is the next one. And that'll be covering the history of life from early life to the origin of eukaryotes. So stay tuned for the next one. Well, I guess don't stay tuned. You have to start like a whole new lecture video, but it's besides the point. Watch the next one, lecture 4.2, and we'll see you later. Thanks. Bye.